Dear friends, good morning to all. Today we can discuss about basic structure of immunoglobulins. Immunoglobulins, otherwise called as antibodies. Immunoglobulin main function is act as antibody. They are antigen binding protein. Antigen binding protein means any infectious condition, any pathogen enter in your body after that which binds to it. Immunoglobulins are present on B cell membrane and also secreted by plasma cells. After secretion, the antibodies circulates in blood and serves as effector of humoral immunity. Any infectious condition, two types of immunity is there. One is cell-mediated immunity, another one is humoral immunity. Cell-mediated immunity means T cell response. Humoral immunity means B cell response. B cell means here the antibody response. Next one, structure of immunoglobulin. Generally, the immunoglobulin consists of two types of chains. One is heavy chain, another one is light chain. Here the immunoglobulin structure, two heavy chain, two light chain. In center, two long chains are called as heavy chain, outer two called as light chain. The heavy chain and light chain, top portion means toppermost portions are amino terminal end, bottom portions are carboxy terminal end. Amino terminal means NH3, bottom portion carboxy terminal for COO minus groups. Two heavy chains are outermost toppermost portions are variable region, either heavy chain or light chain. Both the chains are topmost portions are called as variable region. Bottom portions, remaining bottom portions are called as constant region. The heavy chain consists of one variable region, three constant region, CH1, CH2, CH3. As like light chains have toppermost portions are variable region, bottom portion only one constant region. All the heavy chains and also light chains will be linked with the help of disulfide bonds. And also in heavy chains, CH1 and also CH2 groups in between Heng region, the dark portions are called as Heng region, which is the flexible one region, and also more amount of lysine amino acids are present. And both the heavy chains CH2 portions are attached with CHO. CHO means carbohydrate binding sites. So that the immunoglobulins are called as glycoprotein. Glycoprotein in the sense protein also attached with carbohydrate moiety. And also the toppermost portion, the variable regions are otherwise called as antigen binding region. Antigen binding region. Remaining portion in heavy chain CH1, CH2, CH3 and also light chain Cl or important one portion, biological activity portion. Biological activity means after binding of antigen antibody complex which induces the immune response or biological activity. And also the heavy chains based on subclasses, for example IgG, IgA, IgM, IgD, based on these classes the heavy chain differ, especially mu or gamma or alpha, delta, epsilon, which is based on subclasses. And also light chain also further subdivided into two types. One is kappa or lambda. This is for all the subclasses having the light chain, either kappa or lambda. Next, basic structure of immunoglobulin. The antibodies are heterodimers. Heterodimer in the sense which have two heavy chain, two light chains. Four chains, and the dimeric form means both are same. Antibodies have a common structure of four polypeptide chains. The structure consists of two identical light chain. Polypeptide about 25,000 molecular weight. This is for light chain. Next one, two identical heavy chains, which is for larger polypeptide chains. Center both the chains are larger polypeptide. Outer two chains are light chains. The heavy chain molecular weights are 50,000 or more. Each light chain is bound to heavy chain by a disulfide bond. Both chains are bonded with each other by disulfide bond, and also non-covalent interaction to form HL. HL means heavy light chains. HL2 antibody structure, two dimeric tetramer form. Dimer of dimer means two heavy chain, two light chain. The exact number and also precise position of these interchain disulfide bonds differ among Ig classes. Ig means immunoglobulin classes and also subclasses. Already I told immunoglobulins have 5 types: IgA, IgM, IgD, IgE. These are based on this type, the exact structure which are differ one. Next one, variable region. Variable means the first 110 amino acid of N terminal of either light chain or heavy chain varies greatly among antibodies. The topmost portion, amino terminal ends, 110 amino acids are varying one. These segments of heavy, highly variable sequence are called as V region. VL in light chains. VL means in light chain variable region. VH means heavy chain variable region. In this way, topmost portion variable light chain, variable heavy chain. Next one, most of the differences among antibodies fall within areas of V regions called complementary determining regions. Complementary determining regions means otherwise called as CDRs. CDR means complementary determining region, which is for antigen binding sites. The CDRs of both H and light chain constitute antigen binding site. Next one, constant region. The region of constant sequence beyond variable region is called as C region. Below the variable region portions are called as constant region. This is also two types: CL in light chain, CH in heavy chain. In light chain only one constant region. In heavy chain CH1, CH2, CH3. In this way 3 portions there. Next one, Heng region. The gamma, delta, alpha heavy chains contain a peptide sequence between CH1 and also CH2. The CH1, CH2 domains in between dark portions there. The dark portions are otherwise called as Heng region. It is rich in proline residues. Proline amino acids more amount present. Is flexible one, giving IgG, IgD, IgA segment flexibility. These 3 types of immunoglobulins, flexible one. Next one, immunoglobulins are glycoproteins. Immunoglobulins are glycoprotein means the CH2 region carbohydrate CHO binds to it. So proteins attached with carbohydrate moiety, so called as immunoglobulins are glycoproteins. Carbohydrate attached to C region. in brief digestion of immunoglobulin. For example, IgG with the papain, which produces the three fragments. Those fragments, the two identical fragments, each with molecular weight of 45,000, had antigen binding activity, so called as FAB fragment. FAB means fragment of antigen binding. Next one is the other fragment, totally three fragments, two fragments are antigen binding fragment, another one is molecular weight ranges for 50,000, was found in crystalline during the cold storage. We are maintaining the cold condition, which are crystalline form. It is called as FC fragment. FC means fragment of crystallizable. Next, move on to types of immunoglobulin. 5 types of immunoglobulins there: IgA, IgD, IgE, IgG, IgM. The first one, IgA, found in mucous, saliva, tears and breast milk, which are protect against pathogens, which are dimeric form. Next one, IgD, the part of B cell receptor which activates the basophil and also mast cells, which are monomeric form. Next one is IgE, protect against parasitic worms and responsible for allergic reaction. This is also monomeric form. Next, IgG, which are secreted by plasma cells in blood, able to cross the fetus through placenta, mother to fetus. This is also a monomeric form. Final one, IgM, may be attached to the structure of B cell or secreted in blood, which are responsible for early stage of immunity. Early stage of any infectious condition, IgM only first produced, or which will involve the responsible. This is also the pentameric form. The IgM only the pentameric structure of immunoglobulin. Thank you, friends.